Hello everyone. Welcome to all in the second lecture of the course Fundamental of Agronomy. Today's topic is seed. In this lecture, we can discuss about the different types of seeds, its structure, and the different classes of the seed in the production system and the tags of the respective classes. Now we discuss about the seed. Seed is a vegetatively viable propagule or a fertilized ovule which give rise to a new plant. It consists of three major parts, that is embryo, endosperm and the seed coat. When we call about the embryo, in a mature seed, the diploid embryo develops from the fertilized egg or the zygote. And this particular embryo consists of epicotyle. Then epicotyle is a teeny shoot from which entire plant shoot develops. The growing tip of that epicotyle is known as plumule and a hypocotyle. That hypocotyle is a transition zone between rudimentary or the little root or the shoot. In that part, radicle. Radicle is known as embryonic root and one or the two cotyledons. Then if the seed contains one cotyledon, then it will be called as monocot. And if it has two cotyledons, then the seed is called as dicotyledon. Then major part, the endosperm. The stored food support the embryo during seed germination. This endosperm is fully responsible to supply food until the plant is capable for the photosynthetic activities. Then the next is seed covering or seed coat. It is maternal in origin. It covers and protects the seed from different parts and the mechanical injury. The seed coat is usually hard, thickened and with different type of colors that is partially impermeable to water, that it can uh, be imbibe water in future and activate enzyme and goes to the germination process. It prevent excessive loss from the seed and also act as an barrier against entry of different insect pest disease or the parasite inside the seed. This seed coat is responsible for the dormancy also, that helps in preventing the germination in adverse conditions and make it capable for the germination when the conditions are favorable for germination. There are usually two layers of seed coat. The outer layer is known as tester and the inner thin and delicate layer is known as tegmen. Now the seeds. The seeds are two types, monocot and dicot seeds. In monocot, the only one cotyledon is present and the structure of the monocot seed in the shown in the picture. In this, the first part is seed coat, which main purpose is to protect the seed. It is membranous in structure and generally fused with the fruit wall and it is known as hull also. Endosperm is the bulky and stored food. Its main purpose is to supply the food material or the nutrition to the developing embryo as well as the developing seedling. Alluron layer. It is a proteinous layer which separates endosperm and the embryo. And then in the embryo that is situated in the groove at the one end of the endosperm in which the developing root and shoot occurs. Scutellum. Scutellum is the one large and shield shaped cotyledon. Embryonic axis in which plumule and the radicals are at two different ends exist. Coleoptile and coleorrhea. As the coleoptile means the developing uh, plumule and the coleorrhea is the developing root. Now the dicot, it's mean the seed which contain two cotyledons. Then the different parts are as seed coat, the outer covering. The one outer is known as testa and the inner one, the delicate one is known as the tegumen. Hilum. Hilum is a scar on the seed coat through which the developing seed was attached to the fruit. It means the transportation of water as well as nutrition take place from the hilum. Micropyle, the small pore present above the hilum which helps in absorption of water at the time of germination. Embryo consists of embryonal axis and the two cotyledons. Cotyledons, as we know, are the often cotyledons are often leafless and full of reserve food material that supply the nutrition to the developing radicle and plumule. Radicle and plumule are present in the opposite directions of the embryonal axis, and the endosperm is a food storing tissue that helps in the supply of nutrition to the developing embryo or the radicle and plumule. Now we discuss about the different classes of the seed. The first one seed that is known as breeder seed. The seed or the vegetatively propagated material is directly under the control of the breeder, sponsoring breeder, or the institution, which is the basic seed for the recurring increase of foundation seed. Second class is foundation seed. It is the progeny of breeder seed. The seed stock handles so maintain the specific identity and genetic purity, which may be designated or distributed and produced under the careful supervision of an experimental station, research station, or the university. This seed, this seed is the source of all other certified seed classes directly or may pass through the registered seed. Next one is registered seed. This type of class is not in Indian seed production system. The registered seed is the progeny of foundation seed. It is so handled to maintain its genetic identity, purity, and approved and certified by the certifying agency. It should be of so good quality suitable to produce certified seeds. The most important is the next is certified seeds. It is the progeny of the foundation seed. Its production is so handled to maintain the genetic and the physical purity according to the standards specified of the crop being certified. It should have the minimum genetic purity of 99% and the certified seed may be the progeny of certified seed provided this reproduction does not exceed two generations beyond the foundation seed and also provided that the certifying agency determines the physical and genetic purity of the seed. Next important class in Indian seed production system, that is TFL seed, truthful label seeds. These seeds are produced and sold by the farmer and private seed companies and are labeled with specific information. Then what is the difference between certified seed and truthful label seed? In certification seed, certification is voluntary, while in TFL seed, it is compulsory. In certified seeds, applicable to all notified kind of seeds only, and in TFL, it is applicable for both notified and released variety. Certified seed, it should certify both minimum field and seed standard, and in TFL, it is tested for the physical purity and the genetic purity or the germination purity. In certified seed, seed certification officer, seed and inspector can take samples for the inspection, while in the TFL seed inspector alone can take the samples. Now, some more information about the different classes of the seed. The nucleus seed will have no specified tag and the quality is maintained by the concerned breeder, sponsoring institution or the breeder himself. The next is the breeder seed. The yellow, golden yellow tag is specified for this seed, and the monitoring breeding inspection is the done by the concerned breeder, sponsoring institution, or the seed technologist. The next class is foundation seed with white tags. The State Department, NSC, state and seed corporation and private sectors are responsible for the maintenance of the quality. Next one is certified seed. As your blue tag is given for this type of seed, and the next one seed, that is TFL seed, opal green tag is specified was for this type of seed, and farmer and other cooperative societies can produce this type of seed. Today, this is all about the seed structure, class, and the different tags used in the seed production system. Stay connected with us for more information. Thank you for joining us.